Antibiotic resistance. What is antibiotic resistance? Antibiotic resistance is the development of insensitivity by the bacteria to a particular antibiotic which has been used repeatedly or over a long time, to which it was once susceptible. The biology of antibiotic resistance can be dealt with biochemical aspects as well as the genetic aspects. The biochemical aspects include target alteration, antibiotic inactivation, efflux pumps and outer membrane permeability, target bypass. The genetic aspects include horizontal gene transfer, which in turn include transformation, transfection and conjugation, and mutations. First, let's start with antibiotic inactivation. The antibiotics, when they enter the bacterial cell, there are certain enzymes within the cell membrane which degrade the antibiotic. Such enzymes include hydrolytic amidases, esterases, ring opening epoxydases. The hydrolytic amidases or beta-lactamases, which cleave the beta-lactam ring of penicillin and cephalosporin antibiotics. The esterases are mainly associated with macrolide antibiotic resistance. The ring opening epoxydases are associated with phosphomycin resistance. Inactivation by group transfer. The enzymes transferases, which are located within the cytoplasm, inactivate antibiotics by chemical substitution, that is the addition of adenylyl, phosphoryl, acetyl groups to the periphery of the antibiotic molecule. As these reactions recover co-substrates from the cytoplasm, these reactions are cytoplasm restricted. The antibiotics which are inactivated by group transfer include rifampicin, aminoglycosides, chloramphenicol, streptogrammin and macrolides. Inactivation by redox process. It involves either the oxidation or reduction of antibiotics. For example, oxidation of tetracycline antibiotics by TET-X enzyme. The TET-X enzyme produces reactive oxygen species which bind to certain positions in the tetracycline antibiotic, thereby oxidizes it. The streptomycin virginate produces a type A streptogrammin antibiotic, digenamycin M1, and it is protected from its own antibiotic by the reduction of ketone group to alcohol group at the position 16. Moving on to target modification, it includes proteoglycan structure alteration, interference with protein synthesis and interference with DNA synthesis. The enzymes which are involved in the synthesis and assembly of the proteoglycan component of bacterial cell wall are the principal targets of inhibition by the antibiotics. For example, how mutations in penicillin binding domain of penicillin binding protein is associated with decreased affinity to beta-lactam antibiotics. The penicillin binding protein 2A in MRSA, that is methicillin resistant Staphylococcus aureus, is highly resistant to inhibition by beta-lactams, and the enzymes that are required for the cell wall synthesis remains active at normal lethal beta-lactam concentrations. From the figure, it is clear that the figure A shows the penicillin binding site in penicillin binding protein. When penicillin binds to the penicillin binding site, it leads to a degradation of enzymes which are involved in the synthesis of peptidoglycan components. Whereas in the figure C, it shows that due to mutation, there is altered binding site for penicillin, and hence it inhibits the penicillin binding, thereby the bacteria is rendered resistant to penicillin. Vancomycin normally inhibit the cell wall synthesis of gram-positive bacteria by binding to C-terminal acyl D-alanine residues. However, in resistant bacteria, there are enzymes such as VAN-H which catalyzes the conversion of pyruvate to lactate, thereby preventing the combination of vancomycin, whereas the enzymes VAN-A and VAN-B leads to the production of D-lactate from D-alanine, and hence it inhibits the binding of vancomycin. And in case of VAN-X enzyme, it leaves the D-alanine component, D-alanine residue from the C terminal, thereby inhibiting the binding of vancomycin, and the bacteria is rendered resistant to vancomycin. The protein synthesis interference. Certain bacteria, mainly certain antibiotics mainly affect the bacteria by the interference with the protein synthesis. For example, macrolides and aminoglycosides mainly destroy the bacteria by interference with protein synthesis. Hence, mutations in 23S rRNA component of the 50S ribosomal subunit is associated with macrolide resistance, as macrolide normally inhibits the peptide bond formation in bacteria. Due to mutations in 23S rRNA, there is a normal formation of peptidoglycan. There is normal formation of peptide bonds between the amino acids. The mutations in 16S rRNA gene confers resistance to aminoglycosides due to the post-transcriptional methylation of thylacine. Due to post-transcriptional methylation of 16S rRNA in aminoglycoside binding site. The DNA synthesis interference. The fluoroquinolones interact with DNA and topoisomerase 4 enzymes, and thereby it prevents the DNA replication and transcription. However, mutations in the structural genes is associated with decreased affinity for the altered DNA gyrase complex. Efflux pumps. They export the antibiotics out of the cell, thereby rendering the concentration within the cell membrane